Hello everyone, welcome back to the PYQ series. I am Dr. Amit Kumar Singh, your guide and mentor for the NEET PG journey. In this video of PYQ series, I'll be discussing about Vibrio cholera. This is a very important topic not only for the PG examination but also for UG examination. Several questions has been asked from this topic too, and this topic basically is important because of the disease and because of the toxin, and also question has been asked from the lab diagnosis. So let's start with the video PYQ number 19 Vibrio cholera. So we'll start with the MCQ. So MCQ, it's about a 10-year-old boy who was brought to emergency room with complaints of constant diarrhea that began 24 hours earlier, and the stood at consumption of rice water. He is not responsive and sleepy. His skin color is weak and his eyes have sunken, which is the most likely etiology. So we'll see the information first given in the question. First is the age of the patient, so pediatric patient. Second is constant diarrhea. Third is the consistency of the diarrhea, that is rice watery, and that diarrhea is acute in nature. And then not unresponsive, sleepy, skin texture is weak, and eyes are sunken. This is the fourth information. The first is patient is pediatric. Second is acute continuous diarrhea. Third is rice watery stool, and fourth is signs and symptom of dehydration. Okay, so these are the information given. So we'll see the option first. Shigella sonii, Vibrio cholerae, Entamoeba coli, Escherichia coli. So if you see the first option, Shigella sonii, if you remember, if you have gone through the Shigella dysentery video, so I have told you that Shigella initially may cause watery diarrhea, okay, but then this watery diarrhea will never lead to dehydration. So dehydration is not a feature of Shigella, okay. So this is out. Then coming to the Entamoeba coli, this also leads to dysentery, so this is also out. Now these two are known to cause diarrhea, watery diarrhea, and that too in pediatric patients. So what is the clue to the answer? The clue to the answer is the consistency of this stool. This rice watery stool is the consistent or characteristic feature of cholera, which is caused by Vibrio cholerae. So the answer to this question is Vibrio cholerae, or the disease is cholera. Okay. So this Vibrio cholerae, how it looks like. So this Vibrio cholerae is slender, gram-negative, curved bacilli. So you can see in this image, this bacilli are curved. Okay, these are curved bacilli, and these are also motile by a single polar flagellum. Okay, single polar flagellum. And what type of motility they have? They have got darting motility. Darting means jerky motility. Okay, so slender gram-negative curved bacilli having darting or jerking motility. And then they are classified, this not this Vibrio cholera, but this Vibrio species is classified into halophilic and non-halophilic. And this halophilic Vibrios are basically those which can tolerate more than 6% of sodium chloride solution, whereas this non-halophilic will tolerate only less than 6%. So though both of them can tolerate sodium chloride, but then the concentration varies. So if it can tolerate more than 6%, it is called as halophilic. If it tolerates only below 6%, it is called as non-halophilic. And one more thing about non-halophilic, that these non-halophilic does not require sodium chloride for their growth. However, this halophilic will need sodium chloride for their growth. So what are those halophilic? Are Vibrio parahemolyticus, Vibrio alginolyticus, and Vibrio vulnificus. Whereas non-halophilics are Vibrio cholerae and Vibrio mimicus. Vibrio mimicus. Okay, so they don't need sodium chloride for their growth. Now the serogroups. So the serogroups, the Vibrio cholerae are divided into certain serogroups on the basis of the O antigen present on the lipopolysaccharide layer, and on the basis of this they have divided into more than 200 serogroups. So among these 200 serogroups, this O1 and O139, these two serogroups are commonly associated with cholera. whereas this rest of this non-O1 and non-O139 are not known to cause cholera but may cause gastrointestinal diseases but not similar to that of cholera. Okay, so this non-O1 and non-O139 does not causes cholera. Now this O1 has got two biotypes. One is classical and one is Eltor. And these biotypes have got three serotypes, that is Ogawa, Inaba, and Hikojima. I'm not going to go into detail of this, okay, because question has not been asked from these biotypes and serotypes, okay. But just for the sake of quick recap, I'm telling this. Natural habitat of Vibrio cholerae is coastal saltwater and brackish estuaries. Why not deep seawater? Because of higher concentration of salt. Okay, so that's why coastal saltwater and brackish estuaries are the natural habitat for Vibrio cholerae. Okay, and the transmission is through ingestion of contaminated water with human feces. So those water which are contaminated with human feces, if the person will ingest those water, will get the disease. And the reservoir is only human, no known animal reservoir for Vibrio cholera. Okay. Now coming to the pathogenicity. The pathogenicity of Vibrio cholera is via cholera toxin. So it is a toxin mediated infection. But cholera toxin is not only responsible for pathogenesis. Apart from that, toxin co-regulated pilus. Okay. Toxin co-regulated pilus, in short known as TCP, it is required for the survival and multiplication of Vibrio cholera. And this TCP, cholera toxin, and other virulence factors are regulated by this TOXR gene. Okay. So this cholera toxin is basically composed of a single A subunit and 5B subunit. And where this cholera toxin acts on, this cholera toxin basically this B subunit binds to the GM1 ganglioside receptor present on the intestinal epithelial cell. Okay. And this then when it binds, it triggers the entry of the A subunit, and this A subunit will act on this GS alpha. What is the GS alpha? This GS alpha is basically G proteins. Okay, is one of the G protein. And what it will do to the GS alpha? It leads to ADP ribosylation of GS alpha uh, protein, and thus in turn it enhances or increases or upregulate the adenylate cyclase, or in short you can say it activates the adenylate cyclase, and which leads to intracellular accumulation of cyclic AMP. This point you need to remember. Cyclic AMP is increased, activation of adenyl cyclase, and thus in turn it leads to accumulation of sodium chloride in the lumen, and thus leads to ultimately watery diarrhea. Okay, so this is the pathogenicity of Vibrio cholera. And one more thing to remember, that person having blood group O positive are more prone to have severe disease. Okay, O positive people are more prone to have severe disease. Okay, clinical feature. So this is the disease of which age group? This is a disease of pediatric age group. Okay, pediatric age group. And the incubation period is less than 24 hours to 5 days. So it is a very acute disease. Okay, and the stool, as I told you, is the characteristic stool which we can see in case of Vibrio cholera is rice watery stool. And this what is this rice watery stool? Basically it is colorless watery fluid with flakes of mucus on it. Okay, and it is non-bloody and non-bilious. Okay, so there is no blood and no bile present in this stool, so it will look like this. It is watery fluid with flakes of mucus in it. Okay, and it has got inoffensive and sweetish odor, and it is basically a bicarbonate rich isotonic electrolyte solution. Remember this, the stool which is seen in case of Vibrio cholerae is basically bicarbonate rich isotonic electrolyte solution. Okay, and obviously if this much amount of water will lose from the body, what it will lead to? It will lead to hypovolemic shock, and the feature of this hypovolemic shock depend upon the volume of fluid loss. So if it is less than 5, it may be less than 5%, it may be between 5 to 10%, or it may be more than 10%. So if it is less than 5%, it will only leads to development of thrust, okay, and the body weight will remain normal. But if it is between 5 to 10%, it will leads to, first, remember, postural hypotension, second, weakness, third, tachycardia, fourth, decrease skin turgor. Postural hypotension, weakness, tachycardia, and decreased skin turgor. And this can be seen in this GIF. Okay, you can see here. Yes. So if you see, the skin turgor is decreased. Okay. And if it is more than 10%, what we can see, oliguria, weak or absent pulse, sunken eyes, and in case of infant, we can see sunken fontanelles, as you can see in this image, sunken fontanelles. Okay, and also we can see wrinkled skin, and then somnolence or coma can be seen. Okay, so these are the findings which we can see in case of hypovolemic shock of different concentration of loss of volume. So how to diagnose a case of cholera? So the lab diagnosis will depend upon direct demonstration of the organism, maybe on the culture and maybe on the blood reports. Okay, so the microscopy, as I told you, it is a cylinder gram negative bacilli, curved bacilli with darting motility. But is that enough? No. What we need to do, we need to inhibit the motility by using specific antisera. Then only diagnosis can be made. Okay, and this can be done by using dark field microscope. So inhibition of darting motility by specific antisera is the microscopic demonstration of Vibrio cholera. Or okay, and culture, if you see, the transport medium used to transport the specimen are this Venkatraman Ramakrishnan medium and this Carey Blair medium. Okay, so these two are the transport medium used to transport the stool for culture of Vibrio cholera. Okay, apart from this, we can also use enrichment medium while transporting, and the commonest enrichment medium which is also used as a transport medium is alkaline peptone water. Alkaline peptone water. Okay, and the selective medium which is used for culture are TCBS agar and Monser's GTTA. What is this TCBS agar? TCBS is thiosulfate citrate bile salt sucrose agar, and Monser's GTTA is Monser's gelatin taurocolate trypticase tellurite agar. Okay, and also it will show a positive string test. So we will see in this image. So this is a thiosulfate citrate bile salt sucrose agar. Remember, this last S is not for bile salt, this is S for sucrose, and this Vibrio cholera is able to ferment this sucrose and leads to formation of yellow color colony in TCBS agar, and this is why it is selective for Vibrio cholera. However, this Vibrio parahemolyticus is not able to ferment sucrose, and the colony will remain green in color of the medium. Okay, so in TCBS agar, Vibrio cholera will give yellow color colony because of fermentation of sucrose. Okay, and then this positive string test. So what we will do, we will make a suspension from a colony of Vibrio cholerae, and then we will add to it 2 drops of 0.5% sodium deoxycholate solution, and then we will draw the loop away from the suspension, and we will see a formation of a string. Okay, and thus Vibrio cholera will show a positive string test. Okay. Now, apart from this microscopy and culture, we can see certain blood findings indicating Vibrio cholerae infection. First is elevated hematocrit, which is obviously because of dehydration. Then mild neutrophilic leukocytosis, increased blood urea nitrogen and creatinine, markedly reduced bicarbonate level below 15 millimole per liter. If you remember, as I told you, the solution is bicarbonate rich isotonic electrolyte solution. Because of this, in blood report, we will find a markedly reduced bicarbonate level and an elevated anion gap. Okay, so these are the uh, laboratory findings which we can see in case of Vibrio cholerae infection. So I hope I was able to explain the topic well to you, and you will get some benefit from this video. And if you will make your own notes and will revise frequently, you will be able to answer the question in the examination too. So all the best. Take care. Bye bye.